Aloha mai kakou. So I'm Tai. I'm in the Department of Tropical Plant and Soil Sciences, and I'd like to thank you for the invitation to talk today about soils. We've been talking a lot about soils. I changed the title of the talk a little bit. I wanted to focus on the instrument that we developed to measure soil health. And then I wanted to show some research findings that we have on relating soil health to a disturbance gradient, as well as different management practices. And then we'll talk about microbes, because I know that's what you all really would like to learn about. Okay, so before I begin, I would like to first acknowledge the team that I'm part of. So I'm part of a pretty large team. There's multiple PIs. So you can see Doctors Jonathan Dienick, Susan Crow, Mikey Kantar. Some of these names familiar. You recognize some of these faces. New, Dr. New's also here, as well as several technicians. So we have our lab manager, Christine. We have India Quatin, Destiny, Mitch, Alec, a lot of people. So I'm going to show you a lot of work that has been performed by a lot of people. This was a very much a team approach, as well as postdocs and many graduate students along the way too. So I want to start by asking you, okay, so I have 8 different photos here of 8 different scenarios. So we have different land uses, we have farms, we have forests, we have pastures. We also may have different types of soil. So let's say our goal is to characterize soil health. So the first question I have to you is, can you measure soil health? Can you measure soil health directly? If I asked you, can you please go out and measure soil health? What might you do? Any ideas on what you may do to measure soil health?
Measure some soil chemical properties and some physical properties.
Okay, so you can take a soil sample, perform some laboratory analyses, maybe in-field analyses, and you can measure different properties. Okay, so are you measuring soil health directly when you do that? No. So what are you measuring in that case? We may call them indicators of soil health. And so you may go out, come up with a list of soil health indicators, measure them. But how do you think it would compare across different systems?
I have a question. Maybe it's kind of philosophical, but how do you define soil health?
I'm going to define that in the next slide.
Okay.
Great question. But different systems, do you expect different systems to have equal values of quote unquote soil health? No. Which of these pictures may you predict would have the highest soil health? Would you expect higher soil health in a forest, in a pasture, in croplands? Any gut reactions? What about across different soil types? Do you think all soils are equal in the sense that one soil that's very different than the other has similar soil health? Okay, that's good, because that's what this talk and research is trying to unravel here. So you may want to think about, maybe write down if you have any predictions. Maybe I'll give you a moment. Do you predict forests, pastures, croplands that have higher soil health? And think about what we may measure to determine soil health. So first, yes, we're going to start with a definition. So we do this in our own group, whether I'm talking with soil scientists that are looking at soil health and soil fertility, whether we're writing papers to a general or to our audience or to our peers, we always start with the definition. What do we mean by soil health? Because this can be a pretty abstract term. So how we define soil health is listed here. So this is a definition that's been built over 30 years, which is the capacity of soil to function as a vital living system. So we're here to learn about microbes, right? Microbial activity is part of soil health. It's living. To sustain biological productivity, maintain environmental quality, promote plant, animal, and human health. That's a big definition. That's trying to do a lot here. And so we can think about, okay, we're trying to measure soil health, but in that definition, it's pretty layered. So the words that we used, how we described it, spans from objective ways of looking at soil health to subjective ways. Also, very quantitative ways that we can approach or look at it, as well as qualitative. And so we're working at these different layers where we can talk about soil properties. So Xiaoding mentioned, oh, well, we can measure the physical, the chemical. biological properties. But I also in that definition talk about functions. So what about biodiversity, food, water, air quality, soil fertility, yields? How does it function? And then also what sort of societal value do we add? In that definition, we did include human health. And so soil health is operating at all of these. But here, to start with, we may think about what Xiaoding mentioned with what can we quantitatively measure to give us an idea of a starting point of soil health. So as mentioned, soil health is part of physical, biological, and chemical properties. And so if you've taken a soil science class, then you've probably learned about physical properties, which can include soil texture, structure, porosity, bulk density, how water is retained and moves in soil. Biologically, of course, we have microbial abundance, diversity. We're going to talk about diversity later, nutrient cycling, symbiosis, disease suppression, and chemical. So we may want to assess the soil pH, electrical conductivity, nutrient availability, the ability to retain and supply plant nutrients, organic carbon, as well as toxicities. So we can fill in, okay, these are measurements that we can take on a gram of soil. And then how are we going to determine some soil health measurement? And so as we mentioned, these measurements that we may take here are soil health indicators. And so these are empirical measurements that we're taking, but really what we want to do is try to come up with a score or an assessment of a theoretical construct. And that theoretical construct is soil health. And we operationally define that by measuring these indicators. And so the approach that we've used is to employ structural equation modeling. And so to come up with a way to use these indicators to give us some idea of a soil health score, and that being a latent variable that we can't directly measure. And so this method was developed in social sciences, and this is an example. So let's say most of us have flown in a plane before. And so we may think about overall flight satisfaction. How do you measure overall flight satisfaction? Well, you may break those up into two different components. Well, maybe it's the fairness of the policies. We've all thought about the policies and how fair it is from airline to airline, and maybe the reliability. Maybe those are two important concepts that you want to try to measure that make up soil health. And then from that, you still can't necessarily directly measure fair policies or reliability. So then you think about, okay, what are the indicators of fair policies? What are the indicators of reliability? And then that's where you can measure, okay, what are the booking fees? How do they differ from airline to airline? What are the change fees? What's the number of canceled flights, the number of delayed flights, and the occurrence of lost luggage? And so this is the framework that you can use using these indicators then to measure these latent concepts. So we took this approach. And so what we did is over the years conducted soil surveys. And so what I'm going to present in the scoring function I'm going to present is based on over 50 sites across 6 different islands, encompassing 9 of the 12 global soil orders, with and without a history of plantation agriculture under diverse land use. The point of this is that we did a pretty large campaign to try to capture the diversity that you may find, the diversity of soils, diversity of management, diversity of legacy of management, to try to see, okay, well, how do soil properties range? And then from that, then can we come up with some way of coming up with an assessment of soil health, specifically to develop a soil health score that we can then look at impacts of management or impacts of land use. So this scoring system was originally based on 659 samples. Today we have almost 2,000 samples from many different fields and projects. And generally we're looking at the top 15 centimeters of soil. Now we have a suite of analyses. These are chemical, biological, and physical. That's another discussion in itself. Are we measuring the right things by confining ourselves to laboratory measurements? So we can talk about that more later. But what the approach that we took is there's 44 potential indicators that you see folks using more broadly. You can't measure 44. Not all 44 are going to be applicable to here, nor are they going to be sensitive, realistic, feasible, economic. And so we tested all 44 and then selected 9 of the most sensitive, reliable indicators that we can routinely measure. So we call this our minimum data set. So we have 9 soil health properties. And so this is what it looks like. So this is the scoring function that we have, where we have, okay, you can come up with an overall soil health score, and we base this on 3 different factors. And so these factors are loaded by different sets of measurements that correlate with each other, or are related with each other, or they group together. So here we have hot water attractable carbon, soil respiration, mineralizable nitrogen, and water holding capacity. So these group together, and then you can go a step further, say, okay, well, what are these telling us? As a group, is this telling us some sort of information, specifically maybe about a function of soil? And so for this, you can think about maybe it's substrate availability. That could be something as a core theme to bring it together. Now, factor 2, we have water stable mega aggregates, acidity, total organic carbon. So Sebastian and Christy have taken my soil fertility class before, and you all remember this in Unit 1. Oh, and Ben too. You remember this in Unit 1? These are my first 3 lectures. These are the overarching principal elements of understanding soils, the core soil properties. And so these interestingly group together too. And then the last is enzymes. Enzymes they generally are a wild card for us. They sometimes behave differently, but they're going to give us an idea of nutrient cycling, theoretically, some biological activity. Now, what's really interesting about this, though, is what we found, the 3 groups don't align with our traditional conception of physical properties, biological properties versus chemical properties. They didn't split into those scientific disciplines. They cross each other. So you have a combination of chemical and physical and biological within each of these groups. So then we can think, okay, well, as a function, what are these telling us? And are they telling us something different from one another? The other thing I want to mention too is soil mineral class. So I have that at the bottom. And what this means is that we can adjust the scores based on soil mineralogy. So depending on what type of minerals and clays are in the soil, just like you mentioned at the beginning of the talk, you don't expect all soils to be similar to each other. So in order to compare apples to apples and not apples to oranges, we can adjust this so that the different soils have their own scoring function. And so that way it's a more fair comparison. You can also adjust it on land use history too. We've done 3 iterations of the scoring function as we've gone from 6 to 1300 to 2000, and it's actually a pretty consistent, it's a stable and reliable function. So I just want to show you why we might want to adjust for soil mineralogy. You can also look at legacy, previously intensive agricultural land use. That could be something you'd want to adjust for too. These basically provide context. So the soil health indicators themselves are sensitive. The idea is that they would respond to change in management, change in land use. These are contextual. And so basically, they're giving us a framework or context for interpreting the results. You can see here, I'm showing you total organic carbon and the differences. You can see the impact that intensive agriculture, if you have, most of these sites have had over 50 years of intensive agricultural land use. And then you can also see the differences between soil classes. So our high activity, what we call high activity soils have proportionately more 2-to-1 clays, such as smectite, low activity, more 1-to-1 and iron aluminum oxide. And then your poorly or non-crystalline is what you think about in terms of your volcanic soils that have more the amorphous or poorly or non-crystalline mineralogy. So just a snapshot of the coverage where we are up to date. This is all the 2000 soils in our database, where you can see how we have the coverage of different land uses, soil mineralogical class, and then history of previously intensive agriculture. We've attempted to get good coverage, but you do see that we have proportionately more annual cropland. I'm in the Department of Tropical Plant and Soil Sciences. We work a lot on farms. A lot of our projects is looking on farm management with longitudinal sampling. And so we have proportionately more in that. And then our high activity clays too, we have a lot of farms that we're working on under that soil type. So it's a little bit proportionally more. But overall, we feel pretty good about the coverage that we have managed to get. Okay, so before I finish the piece on the scoring function, I do want to share some of the latest phase that we have. And so we are using FTIR spectroscopy to try to do more of a rapid assessment. The measurements that I've provided or listed so far, a lot of them are benchtop, a lot of them require wet chemistry method. It can take up to 8 weeks just to run a set of samples, very labor intensive. And so we have a project where we're looking at FTIR and saying, okay, well, we can perform measurements more quickly and cost effectively if we were to scan the soil. So here you can see, okay, so you come up with spectra. And then what we've done is we've worked with a couple of students, and then Peter Sadowski talked last week, is that right? Or last seminar. So he's working with us on this project. We're using machine learning then to relate that spectra to our soil health score. So we've been pretty impressed with how it's performed. You can see here for the raw soil health score, it's worked well. And then if we want to look at the soil health score in terms of a 0 to 100% rating as well, it's performed relatively well. So for some of our work, if we're working with farmers and doing quick assessments, we're actually using this function to provide soil health scores for our outreach activities. Okay, so I started with a question. What do you think the soil health score is? So if I give you a score from 0 to 100, this is how these soils rated. So we have anywhere from 17 to 77 out of 100. You can also talk about in terms of percent, but this means it's in the 17th percentile. So for instance, I have 2 kids. I go to the doctor, they tell me if they're in the 99th percentile for height or weight. That's the same as this. So we're giving you a score based on the percentile in your peer group. You have 2 functions for that, for boys, girls. They're all 2 different. So they're going to be scored based on the soil class that they fall within. But that gives you an idea. So we generally, I'll show you the next slide, we see increasing scores in perennial systems, but we see a range of scores as well. And so this is what it looks like if you look at over 1,000 samples across different land uses. This is the scores. This is how they've scored out. And so I have annual cropland to the left, followed by our perennial pastures and forest systems. You see there's a big spread. So you see a distribution of samples. So just because you're in one class doesn't mean you're always higher than another land use, but in general, we see increasing trend. So as you move to perennial systems, and we have higher soil health scores, annual cropland. So this is interesting where if you look at conventional methods, intensive agriculture, we tend, not always, but we tend to have lower soil health scores. After implementing organic methods, you see an increase in soil health scores, and then abandoned cropland too. When that land is unmanaged, grasses grow, you see an increase in soil health scores too. I'm not trying to advocate for abandonment of ag land. I'm just showing you that when you do cease production, you can see that rebounding. Okay, so I want to share a few research questions with you and how we use the soil health score to answer some of these questions. And so our first question is, how does soil health change along a disturbance gradient? So I had a student, Christina Estrada, a master's student who worked on this. And so she made a composite indicator of anthropogenic disturbance. So she based this on the European Commission for the EU, and then also looked at ways that they measured anthropogenic disturbance in other systems like karst systems. And you can see here that she had indicators. So she went out and she did a survey and she surveyed land managers. She used GIS to look at some global data. And then she took some soil measurements herself to get an idea of biotic indicators of soil health, such as like removal of biomass, burning of biomass, geomorphological, such as tillage, or she also looked at the compaction as well. Atmospheric, such as ammonia deposition is one. Cultural, so folks understanding, land managers understanding of their own disturbance practices, as well as hydrologic. And so, again, through a series of surveys and questions, she compiled this using that coined indicator from the EU. And we did field campaigns. And so here we, well, Christina and Tanner, I actually did not go on these field campaigns, but you have Maui, Puerto Rico, and Pohnpei, where they went out. So they controlled for soil type. So in Maui, they just looked at andesol, so stands specifically. Puerto Rico, and they looked at ultrasols, humus specifically, and then Puerto Rico, oxasols, peroxides. These are very, very old, highly weathered soils in this case. And then our idea was to go along a disturbance gradient, knowing that she would score within. So there should be a range of disturbances within each of these. And so she looked at vegetable systems here, then low and high elevation pasture systems, and then shrubland. And then also too, in Puerto Rico, looking at yam, plantain systems, then coffee, and then secondary forest. And then in Pohnpei, looking at kitchen gardens, agroforest, and primary and secondary forest. And this is some of the data that she found. So this is soil health score. It is adjusting for mineralogy. Remember, there are 3 different soils, but we have that adjustment in our scoring function. So we're able to compare apples to apples. And you can see that overall, she found a decrease. It wasn't linear. It's a curvilinear decrease as you increase disturbance. But what's really interesting is to bring it together on a single figure, so aggregating the data. What we found here is there was a breakpoint. So she did a breakpoint analysis. This was the best fit to explain the data, where she saw, okay, there was a slight decrease up until a certain point. And then once you get this point, so this breakpoint was 53%, it was actually surprisingly similar across all islands. All islands, it was generally between 45 and 55% they saw, or we were able to see this decrease in soil health with disturbance. And then there was a sharp decrease. So what's interesting here is that these are perennial systems. So the perennial systems with disturbance, you do see some decrease in soil health, but it wasn't as remarkable as with the cropland. But with that said, when you look at the cropland, so the cropland is in yellow here, you still see cropland can be high, particularly under lower disturbance in these systems. So what's happening here? Well, according to her data and her surveys, these samples generally corresponded with high tillage, and as well as management like pesticides and or pesticide or herbicide and insecticides were some of the main factors. Okay, second question. So that's looking disturbance, particularly comparing across different land use. What if we just look at a single land use and say, okay, well, how does our management impact soil health? Can we do some longitudinal studies? So in this case, we have soil health changes in response to compost additions. And so here we have a green immune farm, a mollisol, without a history of plantation agriculture. And I have two panels here. And so on the y-axis, we have change in soil health. And on the x-axis, we have years. And the first panel to your left is control. So this means there's no management change. On the right, we have compost addition. So on this farm, it's a conventional farm, we had plots where we added compost, and you added compost annually. So here you can see the arrows represent additions of compost. So what we found is we're looking at the change of soil health. So each year we saw an incremental increase. So overall, we have a change in about 15% increase in the soil health score. So we're still doing this research. We have it at different farms. We find that this has been replicated at different places, but the response can vary. Sometimes there's also noise too, where you can see up and down a little bit. So it's not as clear in the response. But what this is going to tell us is, okay, what we can tell a farmer is, so you adopt this practice. This is what you may expect in terms of your trajectory, the trajectory of soil health changes that you may expect due to the implementation of this agricultural practice. Okay, so the final question that I'm going to talk about before we talk about microbes is, how does soil health relate to other functions? So I'm going to relate it to soil fertility, but we're also having work where we're performing work where we're relating it to yield and other outcomes too, other field level outcomes, because part of this, I hope you've been thinking about how it's kind of circular. Okay, you're measuring a lot of carbon pools, you add compost, you it would make sense, right, to increase soil health because it's carbon based. So it can be somewhat of a circular argument. And so what we want to do is say, okay, well, what about the other features? What about yield? What does it mean to yield? If you increase soil health, can you increase productivity? We can also think about ecosystem services. What about runoff? So we're venturing into that research of trying to say, okay, the so what? What can you expect of maybe some of the benefits or co-benefits of increasing soil health? Okay, so we're going to talk about soils for a second. So we're going to ask the question, how does soil health relate to soil fertility? And we're going to look at the different major soil classes of Hawaii. So here, I mentioned it before. So these are the three main classes of soil that we included in the previous. So you have high activity clays, low activity clays, and poorly non-crystalline clays. Now, within these, you have a difference or a range you can have in fertility. So for our high activity clays, so here's an example of the Lululule series. You can see the mineralogy right here is classified as smectitic. These generally we think of as more fertile soils. Now, our low activity clay and our poorly or non-crystalline clays, we can have differences in soil fertility. That's related to the mineralogy that you find. So low activity clays, you probably are familiar with those in terms of the color. So who's seen red soils? And okay, so those are generally can be classified here. Now the interesting thing here is folks like to call those infertile. They're not necessarily infertile. We can have oxasols, in this case, that are more fertile than others. And here's an example. So the more fertile one is the Wahiwa series. You probably may have seen, worked with, heard of this soil before, versus the Kapa'a series, which is less fertile. You can see here, this is kaolinitic and this is sesquite, so more dominant in your sesquite oxides. And then poorly non-crystalline, again, you can have more or less fertile. So the soil to the left here, Pula series, this is our ustan. Its mineralogy is amorphic. This is what we often call like a good sweet spot for like peak soil fertility. And then here it's relatively more fertile than our Udans, is the Hilo series. You can see here it's very hydritic. So how, for each of these soils, does soil health relate to soil fertility? So if we were to map this, so here we can go through the same process that we did for soil health. We have a suite of soil nutrients like phosphorus, calcium, potassium, micronutrients that we can measure, go through the same process like we did with the scoring function. And then we can relate soil health scores to soil fertility scores. And then we can say, okay, well, do we have cases where there's low soil health and high soil fertility? Or vice versa, or both high soil fertility and both low soil fertility and soil health? And so we find some interesting patterns here. And this will help us understand, okay, what are expectations when we're working with different soils? And so here's our examples of tour, so vertisols and ustols, which is mollisols, where we see a lot of samples falling in this first quadrant. So we have high soil fertility, but low soil health. These are pretty productive soils. Doesn't mean we can't. I just showed you an example of how we increased soil health in at that green onion farm in the mollisol. But in general, we see them in this upper left quadrant. What's interesting is the opposite is true for our anthesols. So we have the UDAMs here and the USDAMs here. So the UDAMs are interesting because they have high soil health, but low soil fertility, so low nutrient availability. But those carbon pools that we were measuring were quite high. The USDAMs are also interesting because now they're spanning. So in general, they have very high soil health, and they can also have high soil fertility as well. And then to me, this one's even more interesting. These are oxasols, where you see impact of weathering intensity here. So our toroxes, so our drier, if you think of moisturizing, are over here, where they tend to be lower soil health but higher fertility. And then as we move to ustols or peroxes, so that's more rain, also more highly weathered too. We see an increase in soil health corresponding with a decrease in soil fertility. So this is really interesting. It helps us in agronomy and a lot of the circles that I work in, that's the question. So what? How we measure soil fertility? What are we missing if we don't measure soil health? And to me, what this is saying is that you're seeing different things depending on what you're working on. And so how do you bring together this more holistic understanding is a good challenge for us. Okay, so what I'm going to do for the next 20 minutes, I think, is to invite Giovana up to talk about her microbial work. So we have a grant that we're working on, that's the FTIR is part of it, but we're looking at soil microbiome. So Giovana and Christian Palmer are both working on this and developing some pretty neat tools and providing some really great insights into how we can use the microbiome to understand soil health. So I think Giovana presented last year. If you don't know Giovana, she got her undergrad and master's from São Paulo University and then in Brazil. And then like me, she went to WSU for her PhD. She studied agronomy and bioengineering or engineering for her undergrad, and then for PhD worked in veterinary science and translational research. She's been with us for a few years. I can date Giovana because I had a baby right when she started, and so I've known her as old as my son. But I'd like to invite her up and talk about some of the really nice research that she presented last week at Tri-Societies.
Aloha, everyone. Thank you, Tai, for the nice introduction, and thank you for inviting me to be part of this talk. As Tai said, many of the indicators that are used to score the soils are driven by microbial activities and functions. So it has been really interesting, but not very straightforward to answer this question, what microbes are driving soil health. So I gave a seminar here in M4 in February of this year. And if you remember, I show that land use drives bacterial and fungal diversity. So what I talked about was in panel A, we can see bacterial richness, and panel B, fungal richness. And we can see that as land uses change, there's also a change in microbial diversity with mostly agroforestry and orchard systems, as well as unmanaged lands with higher microbial diversity. However, when we look at soil health scores and the relationship with bacterial and fungal diversity, we don't see a correlation. Again, we're looking at bacterial and fungal richness and the soil health score for many samples from these different land uses and using different management practices. And we see that only 1 or 2% of the variation in the data of the scores are explained by microbial diversity. So different from what many people suggest that a healthier soil will have a higher microbial diversity, we are not finding this in this data set. And also, as I present this, I'm also seeing that other groups that also study soil health, they are showing that they're not finding this association between soil health and microbial diversity. So there is this paper that is out for a while. The title is How Microbes Can and Cannot Be Used to Assess Soil Health. And the authors, in a very elegant way, they discuss how higher soil bacterial diversity is not always ideal. So the same way we could have a higher diversity of what can be considered beneficial bacteria, we could also have a diversity of potential pathogens for plants, for example. And we don't want that. And also, just as human beings have different gut microbial communities, but they still can be healthy people. For the soil, it's the same thing. It's very complex, but there's no ideal soil microbial community. So with that, we found that microbial diversity cannot be used as an indicator for soil health. And one of my takeaways when I presented the seminar is that most of the traditional molecular tools that we use, so analysis such as alpha and beta diversity, cannot be used to predict soil health. So instead, how can we use microbes to predict soil health? So we've been thinking about this for a while, and this was inspired by many of the human studies where they look at gut microbiome and try to predict health outcomes. The same for soil and humans, very complicated to define health, because for humans, for example, the absence of disease is considered health. And this definition is very challenging. However, what we thought about is that we could think of it as a soil microbial health index. So by looking at the microbial composition of the soil, we could calculate the degree to which a soil sample portrays the bacterial genera associated with either high or low soil health scores. So for one sample, we'll calculate a number without a unit, similar to a scoring function that will tell us if the microbial community of that one soil sample resembles a community of samples that have high or low soil health scores. So this idea is still being developed, and we're working on it. I'm going to talk a little bit about how we calculate, but it is still not published. So if you want more details or if you have any questions, feel free to email me, but I'm going to show what this looks like. So we use 293 soil samples collected from annual cropland systems in Hawaii, systems with different management practices, such as conventional, organic, system with different soil types, with different crops grown. We wanted to also create this indice to represent the agricultural systems here in Hawaii. And part of this data was used to construct the model, part of it to validate the model. And to begin with, we needed to characterize the soil samples. So we divided into, even though it's not very straightforward, we divided into high and low soil health score samples. So if a soil had a score bigger than 0.5, this is considered a high soil health score, and lower than 0.5 a lower soil health score. And after we did this characterization, we use differential abundance analysis to find which are the bacteria associated with high and low soil health scores. And we found 82 bacteria genera that I'm going to talk about in the next slide, which are calling biomarker for these two sets of samples. And then the way that we calculated this index was not only looking at the richness to know like which of these bacteria are present in each sample, but also looking at the geometric mean of the relative abundance, taking a ratio of these two proportions, because this will represent how similar a soil sample, the microbial community of that soil sample is to other soil samples that we have with the different spores. So here's what we found. We found this 82 bacteria genera that we are calling biomarkers. And on the top, we found 43 that were associated with low soil health scores and 39 bacteria that were associated with high soil health scores. I'm not going to talk too much about this, but one interesting thing that we found is that some of these bacteria, such as Salvibrio, Pseudomonas, Devolia, Bridorhizobium, Spingobacteria, there are bacteria that are normally found in soil samples, but in the literature, they're also known to be associated with fungi, and they're part of the group that are associated with high soil health scores. And just before I talk about the results of the index that we created, once we divided the data into high and low soil health scores, if you look again at diversity, we can see that does not stratify the data well. This we have for both bacteria and fungi, we see no difference across these two data sets of samples. However, once we calculate the soil microbial health index that I just presented the concept, we can see that this stratified the data, and we got this difference showing the different microbial communities in these two sets of samples. And then the next step was to look at soil health indicators. So first, we look at how the soil microbial index for each of the soil samples correlates with CO2 bursts, so soil respiration, and we can see that they correlate. However, we also look at total organic carbon. There is a weaker correlation between the soil microbial index and the total organic carbon, but we can see this tendency that as you have a higher soil microbial health index, there is an increase in the CO2 burst and even the weaker increase in the total organic carbon as well. And then finally, we look at the correlation between the soil microbial index to see if we could predict the soil health scores that Tai was talking about before. And we also saw this correlation between the index and the soil health scores. And if you remember, Tai just showed the data about applying compost in this specific farm and how the soil health scores increase with this intervention. And we are also interested in looking at the microbial community. So what happens once we apply compost? So we look at samples that receive compost regardless of the rate or frequency. And what we found that as we saw the shift in the soil health scores that had a similar pattern with the soil microbial index, but what we saw is that many of the bacteria genera that are enriched in the compost samples, the samples that receive compost, they are part of the high soil health score. So the genera that we use, that I presented before, that we use to create the index, and samples from the control treatment, they had this higher enrichment of bacteria that are part of the lower soil health score group. So this is interesting because we can see this connection between the scoring function, an intervention, so a change in management in the farm, and how it can also affect the microbial composition of the soil, and also the changes in the index. So here's another example, like Tai showed the different farms with the different soil health scores at the bottom. On the top, I'm showing the soil microbial health index. So this have in our concept low or lower soil health scores. And then we can see this negative microbial score. I forgot to mention, but the way that the score is calculated, if it is positive, it means that the microbial community present in the soil sample is more similar to the ones that are present in the high soil health scores, and vice versa for if it's a negative value. And we can see how this changes as the soil health score also changes. And then we're really interested in looking at agroforestry, orchards, pasture, other systems as well. That's it. Thank you very much.